amazing things right, especially the snare protein, the protein that really promote fusion. So there's now in the iBio seminars an interview that the scientist that started the iBio seminars, Ron Vale, you're going to be watching one of his videos also. He works on the cytoskeleton and motor proteins. He interviews them together, and he asked Jim to describe Randy's work, and he asked Randy to describe Jim's work. And you kind of see that at this point, after Nobel Prize, they are the best of friends, but there was quite a bit of tension while this was evolving. And what is really important is because they basically answered these two major questions, right? We've been listing the problems that cells have to solve. And two of the major problems are how these membrane vesicles that were known to exist, they could be seen on electron micrographs, how are they budding? And after they bud, how they fuse with the next acceptor part. So there was some confusion. This means maybe I should clarify. Some people were asking, why am I asking you guys to put your quiz of the videos one day before? Is to give me time to look the answer, so I can evaluate, right? This means, is there some confusion? And everybody did quite well, except that there were some important misconceptions about what the snares do. So snares are very important class of molecules, and they are very different from proteins that promote budding, right? There's some confusion because after the vesicle buds, the snares are there to promote fusion, but they are not coat proteins that deform the membrane at all. What they do is that they bring the membranes together, right? So this means this is the classical concept that emerged out of Jim Rothman's work, that one is anchored on the acceptor membrane, another one is anchored on the target vesicle, and there's actually one, this snap 25 or 23, there's several snaps, that forms the third coil-coil bundle, helix, that it is the bundling together of these vesicles that is believed to generate the force to really promote the fusion. So this is what is drawn here, right? That these are the steps of fusion. That you have, of course, hydrophilic forces kind of repelling these membranes, and then when the snares engage with each other, they start to, so it's as if you are twisting a wire and you're bringing the membranes together, and then this overcomes the water molecules, which are pushed out, and you get the first step of fusion. This is the classical Henny fusion, in which two membranes fuse, but the ones in the middle are still not fused. And then there is the complete fusion. So people have reconstituted this in vitro, and the brilliant finding of Jim Rothman is that he managed to purify these proteins by using some of the proteins that bind to this complex as a bake-off. And then the other very smart thing he did is that he used extracts of brain, which have a lot of these synaptic vesicles that have to have this machinery. So he basically went straight to brain, a mammalian brain, to purify this, while Randy Shackman was working out the genes by these screens in yeast that you heard about. So what emerged is an amazing explanation for the biggest mystery of all. How does the cell determine what goes where? So this slide here looks very complicated, so nobody has to worry that you're going to have to memorize which snares are in which compartment. We're not going to ask you this, so we're looking for big picture concepts, right? What you have to know is that there's always a T snare on the target vesicle, a V snare on the acceptor molecule, and you have different ones. So this explained that when they started to purify these organelles and see which types of snare molecules were present, they represent the signature of these. So they are address labels, basically, that prevents membranes to fuse with the wrong one. And that's not the only mechanism that cells use. There are other ways to prevent membranes to fuse in the wrong place. This is something that cells put a lot of energy into because specificity is critical. Right? So the interesting thing in the history of the snares, right, both paths, right, East genetics or the biochemistry done in the Rothman lab, they had to work with what they could handle, right, experimentally. And really a strike of luck in Jim Rothman's lab is that they found one protein which was sensitive to this agent here, ethylmelanine. So they could inhibit it, and then they could start reconstituting, and they would know when they needed this factor back because the one that was already there was not working. So they started with this, and it turns out after a lot of work that actually this protein, for a while there was a lot of confusion and a lot of people accused Jim Rothman of getting it wrong because at one point he suggested that this would be a fusion protein. So it's not the fusion protein, but it's essential for breaking the bundle after the fusion happened. And it's an ATPase that breaks this complex and releases the snares to be used again. So it's essential because if you start not being able to break down the complexes that form, you run out of snares very fast. So that's why this emerged, and this was used in SF and the accessory proteins that interact with it as a bait to fish out the snares, because they postulated this was the really important conceptual advance that they made, that they imagined that to promote fusion, you need membrane anchored proteins, right? You need something to be tethered to the membrane very strongly to push it. So they made this hypothesis. Well, we're going to use these soluble proteins then to fish out and look at what is a membrane protein based on the hydrophobic data, right? And these snares have this terminal that we kind of looked at how these proteins that have only a short hydrophobic tail get anchored on the membrane. And this is how they brought it out. And when they got it, there were other people working on the systems that had already arrived at those proteins. For example, was important for neurotransmission. So everything came together, and it was a flurry of papers, which was an extremely exciting time. And then along this time, and especially through the work also of Randy Schechtman and other labs that were doing East genetics, another class of proteins that is important for you to know, right? So we're going through snares, very important, right? So snares promote fusion. And then RABs started emerging. Then in many screens, these small GTPases started emerging. And again, they seem to be associated with very specific organelles. That each organelle had a type of RAB that was bound. So this took a lot of work, a lot of people. Peter Novick, that was the student in Randy Schechtman's lab, continued working with this. And what emerged is a field that is very active until now in cell biology, which is how RABs, so RABs are not the fusion proteins, but they help bring the vesicles close to their receptor membrane, right? So you can see here in this diagram that this means, again, this is another common theme, that using the energy of GTP hydrolysis, that what they do is that these effectors of these RABs tether the protein. So once you get RAB GTP associated with the vesicle, they can bind to this tethering protein, and this kind of ties the vesicle close to the membrane, allowing that by chance the snares to target each other. So this is another mechanism, basically to fish out the vesicle and hold it there, and very specific. Different RABs are working on different membranes. Right? So again, so RABs are also not membrane sculpting proteins, and they're not fusion proteins. They are proteins that help with the tethering of vesicles to membranes. Right? They're kind of thought of as what gives the identity to a compartment is the collection of RABs that are associated with it. Let me just show you a little movie here. That this is one of the RABs that is on endosomes, for you to see a nice example of membrane fusion. In this case, it's RAB5 that is fluorescent.
In these cells, fluorescent RAB5 protein has been overexpressed. RAB5 binds to endosomes and promotes their fusion with one another, thereby increasing the steady-state size of individual endosomal compartments. actual membrane fusion effects can be observed at higher magnification.
And this has now been reconstituted. People actually can do this in the East vacuum. Actually turned out to be a great system for these membrane fusion assays. The East vacuum is this giant aerosol that the yeast cells have. So some labs have purified these vacuums and they put whatever proteins they want to see if they promote fusion. And they have a way of reading out. They put different molecules inside and they watch when they emerge. So this, what we talked about until now, right, is this concept of what promotes fusion. It's the snares at the end, the ones that do the last step, and the revs help bring the vesicles to the membrane. But another extremely important step, and what is fascinating in this process is that because of the yeast screen, and they were not discriminating what they were looking for, right? Because anything that was getting in the way of the vesicles reaching the membrane. And the videos, I think everybody understood well that very elegant double mutant strategy that they made to see what came first. So they figured out that the mutations that were affecting secretion that were earlier in the pathway were probably the ones promoting budding. So in this case, you had to think that it has to be something that can deform the membrane to make a perfectly flat membrane, then transform into a vessel. So we're going to look more into this. One of the best understood examples of this is clathrin in endocytosis. I think many of you already heard about the clathrin-mediated endocytosis. So I'll just mention, just because we talked a lot about lipid rafts, these cholesterol-enriched patches on the membrane that you also enriched in sphingolipids. So there's one type of membrane invagination that this protein caveolin is very important to maintain the shape of these structures. And it's now known, actually, caveolin was discovered first. There's a second type of protein that are called cavids. They were just discovered more recently, that they basically create these invaginations on the membrane. And you end up with these structures that are called caveola. And this is something that is still evolving. We're doing some work in my lab on this, and there's still quite a bit of discussion. It's interesting to see which stage, right, that fields are. So people still disagree if caveola just sit on the membrane like that. They are just these, some people think they are reservoirs of membrane. So these proteins, they form the membrane, make it invaginate, but they sit there. And then if the cell suffers some sort of mechanical stretch, they are there to provide a buffer. So there's some nice data suggesting this. There's other data showing that they actually can pinch off and move into cells. And when you look at the inside of the plasma membrane in cells that have a lot of caveola, you can see that they have this structure that is, we're going to see next week, that each coat kind of has a pattern of its own. So the caveolin makes this kind of striped pattern. And this is kind of the fingerprint. Actually, there's a nice example here, just next to it. There's a clathrin lattice. So this is the better study, very well understood how this type of culture associates with it. This is one of the main constitutive pathways of endocytosis in cells. And this other one is there, but people still debate if it's just sitting there or if it's really with the right stimulation. Now, if we go back, right, so what is promoting this important budding of proteins out of the ER? So here for comparison, right, this would be again clathrin vesicles that are mostly involved in endocytosis, but also in other steps, steps. And then you have the COP1 and COP2 that I think many of you answered correctly the questions. That COP2 is a little counterintuitive because it's the order in which they were discovered, right? COP2 is basically coming out of the ER, and COP1 are the ones that are going back to the ER. And they have different period codes. this is the diagram in which you have, right, in green is clathrin, showing that clathrin is in endosomes, but it also has been discovered that in the beginning people were hoping it was going to be all very neat and clean, but what is beginning to show up is that clathrin can be involved in many steps, including budding out of already formed endosomes or even in vesicles in the Golgi use clathrin. And the COPs seem to be really more dedicated to just this system of carrying stuff out of the yard and then sending it back. I don't know if everybody remembers, right? This is when we were looking at signals that help proteins in compartments. So does anyone remember what happens when, so this is already indicating, right, that this sequence KPL on proteins is called an AR retention signal. So how does this work? The slide gives half of it, right?
If a protein that is resident to the ER escapes the ER, it'll have this signal, KDEL signal that is recognized by membrane proteins that go back to the ER.
So where is this happening? Where are the receptors, the majority of the receptors for KDEL located? If they are, right? What you said was right. They are taking proteins that escaped out of the ER.
Aren't they just going to be in ER? A majority of them?
If they are already in the ER, they would never escape.
Should be in the Golgi.
They should be the most. This means, of course, when the vesicle comes back, the vesicle brings the receptor back, right? So you always have to think of the steady state. But so this is the situation, right? So this means if you look here, what's represented as the receptor, this ring, it's a lot of it is on the cis Golgi. So this means that when proteins that have this signature, that they belong in the yard, they reach these compartments of the Golgi, the receptors recruit COP1 and they are sent back. And this can, it's believed to happen mostly on the cis Golgi, but the receptor really can be all the way down and still functioning in shuttling these proteins back. So this was also something very interesting when this started to emerge, because in Jim Rothman's first in vitro reconstitution assays, first he thought he had ER and Golgi, and what he was seeing in terms of vesicle was this transport here. And this caused a lot of arguments because Randy Shankman knew that because of the order of his mutants, that there were proteins that were dedicated to come out of the ER, and they were different from the proteins that so Jim Rothman basically had COP1 and Randy had COP2, and they couldn't agree what was doing what. And at the end, all of it was explained by the fact that Jim Rothman's lab actually did not have ER in their population. They were looking at Golgi to Golgi transport. Because it's very difficult. They had to separate these compartments from cells by density gradient. And they all these large flat cisterns that it's very hard to separate one from the other. So scientists spend a lot of energy kind of criticizing each other and saying that it's completely wrong, and nothing is completely wrong, unless it's made up. So usually it's the interpretation that at the time is not correct. And then with more data, it falls into place. So Randy explained pretty well, right, in the video how, so they worked this out. So what was really remarkable about Randy Shankman's lab is that although they still use yeast genetics, he was trained as a biochemist. So very soon when they realized the importance of what they had, they started doing also biochemical reconstitution assays. And it's with the biochemical reconstitution assays that they really worked out. Right, so this means this very important protein, the SAR1, again, a GTPase, right? It's a thing that keeps coming up. You have these small GTPases, and this is, it is the binding of either GTP or GTP that controls if the protein remains soluble or if it anchors on the membrane of the ER. So this this is another very important concept that it happens in almost all the important regulatory membrane traffic events, that the pool of the proteins that you need to do a certain event, they are in the cytosol. So then what you need is something to direct that protein to the correct membrane, and then to promote the hydrolysis of, if this is controlled by DPAs, to then make a commit to work that they are. So this is done through the exchange of GDP on SAR1, and then beautifully they were able to work out the subsequent steps, right? Because they had, you see here that the names of these proteins are still the names that emerged out of the screens. They called all of their mutants SEC. So this SEC23 and SEC24 turned out to be this complex that recognizes SAR1 once SAR1 is inserted on the membrane. And then SEC24 has the ability to, which is a critical step, right? If you're going to bud, you want to bud something that is going to take what you want out of that compartment. So receptors carrying the cargo of interest are recognized by this complex of SEC23 and SEC24. And this is what triggers the formation of the vesicle, because the next step then is to get this scaffold protein in, right? Which turned out to be SEC13 to 31, that forms this, which is really what deforms the membrane. And we're going to see next on Thursday that clathrin works in a similar way. So these proteins have the ability to form a scaffold that is curved, and it pulls the membrane into a vesicle shape. So all this in the beginning was, this was actually worked out after clathrin. So they already had a model that this is probably how it should work. Questions?
Do you be able to differentiate between the 4 different SEC proteins, or just know that they
Let me repeat for everybody. So you're asking if it's possible to what they did to distinguish?
I'm asking if, should we know just broadly that that the SEC 13, 31, 23, and 24 were vesicle proteins in general, or do you need to know if each one of the 4 does?
I keep getting these questions, but instead of just thinking of the exam, right? You guys should think of what is important to know. Think 10 years ahead, right? This means you're already doing whatever you're going to do later. What is important to know? I don't think you have to know the numbers of the proteins, right? But you have to know the right concepts. You have to know that the snares are not membrane deforming proteins. That's what you can get from it. So don't worry, we're not going to be asking you names and memorizing things, whatever. This specific mechanism of the SAR1 is just an example. When something is worked out so beautifully, we may ask to see if you understand the concept that there's a sequence of events, right? But if you get the name wrong, but you get the concept right, don't worry about that. The very interesting conceptual question is what is on this slide, right? Because if you imagine that a protein only can make this kind of curvature, you would, it's something that people know that there are very large molecules leaving the ER. Collagen is a huge rigid molecule, right? So this immediately came up. And this, I remember also that this was one of the criticisms when people started purifying these vesicles, and Randy explains in the video that they could actually generate those things in vivo, and they were all very homogeneous. And then people kept saying it must be wrong, because how do you get something as big as collagen out? Right? So people always get the exception, and they say, well, this thinking is wrong. And it turns out that this scaffold specifically, the SEC1331, is flexible. It has been shown that it can assemble in much larger buds. So there's evidence for this, and there are some proteins that help this happen. So packaging proteins, right? So the answer is that the cell adapts the mechanism. It all depends if you have a type of scaffold that has a rigid size. Clathrin seems to be one of the examples in which it can't be expanded too much. But in the case of COP2 codes, there's evidence that it can be expanded if you have a very large cargo molecule.
In that last slide, the pro collagen.
The previous one?
Right. is that being transported or is that helping form, I guess, the vesicle? It kind of looks like it's pushing it up, but I'm not sure if that's actually just
Yeah,
the
that's a
thing
good
being
question
transported.
that I think it's not solved yet. One thing that is still being worked out is what pinches these membranes off, because if you imagine something's deforming the membrane, you still have a fusion event to happen at the end to release the vessel. And this doesn't happen spontaneously. We're going to see next week that there's one very well understood protein that is dynamin, that kind of coils on the neck and squeezes it.
Yeah.
But it is emerging that there's a lot of budding events in cells that don't use that. So what is abusing? If all budding events need a PJs, like we call or not, and if you imagine something like this, it's a very good point. So this means it's a very large vesicle, right, that has these rigid molecules inside. How is this pinched off at the neck? I don't think that's resolved yet. But the evidence is that large compartments containing collagen that have also the scaffold have been found. Nobody knows exactly. There's a lot of arguments and debates over this. Another very interesting thing that came together when all this was worked out, and then people had antibodies and they had ways to visualize this, that this concept of the ER exit site emerged. Right? So what is really interesting is that it's not everywhere in the ER that these COP2 vesicles bud out of. So you can see on this select micrograph here that they are hotspots in which this is happening more efficiently. And there seem to be different areas of the ER that receive the vesicles coming back and the ones that are fighting out. But of course, it's very different in each cell type. And this is still very much dependent on morphology. But in these, they are clearly ER exit sites also. And then the Golgi, which I think in all these arguments and really vicious fights among scientists and membrane traffic investigators, maybe there's more discussion about the Golgi than anything else, just because the Golgi is so complex and there's still discussions of, for example, if these compartments are fixed in space and then the vesicles just carry things, right, which is the cisternal maturation model that is called, or if they basically, the compartments on the top, they actually were generated from these large ones that matured gradually. So I'll get back to this. And what varies also is the morphology of this. So in mammalian cells, it's very easy to recognize a Golgi because it's like this stack of pancakes. But in yeast, for example, this was kind of part of the skepticism that nobody believed that Randy was going to get anything worthwhile with his yeast screens because you don't see a Golgi like this in yeast. But the issue is that in yeast, the function is there. You just don't see these well-developed stacks. They have multiple little Golgi that if they, then in some of his mutants, actually, I think you saw this in one of the videos, that one of their mutants that slowed down the traffic out of the Golgi, then they saw something that actually started to look like a mammalian Golgi. And this is what developed the concept that these organelles, especially the Golgi, are extremely plastic, and it all depends on the rate of flow of membrane through it. And something else, of course, that has to maintain this structure, so what is also much more recent work, that they are these fibers that are known to kind of extend, that they call Golgi matrix proteins, so they, some of them are called Golgins, and there's important wraps. So there's basically a meshwork that kind of resembles extracellular matrix that you're going to be hearing about soon. And this is associated with the Golgi and is believed to help maintain the structure, at least in mammalian cells. And another very important concept, so in this case again, right, this means we're not asking you to remember exactly which sugars are added in which order, but what is important to remember is that there's very interesting findings made when people started developing these electron microscopy methods that you could put a substrate to a certain enzyme, and they started to see that the enzymes were separated from each other. So there were some that were present only on the cis Golgi, and then others that were in the trans Golgi. And then this was worked out in which this glycosylation starts in the yarn, like we discussed, but it is finished up. So this final additions of carbohydrate moieties to glycoproteins happens in kind of a station-wise as the protein is moving through the Golgi. And this turned out to be very useful for scientists because it's possible to, for example, use enzymes that will cleave specifically some of these sugars. So if a protein is sensitive to that, people know that it already advanced to that station because it got that route. So this is pretty well worked out, and it's basically emerged that the Golgi is a carbohydrate processing station.
For the two different models of Golgi transportation, have they both been observed, or are we trying to figure out which one is true?
There's evidence for both, and I have a slide at the end. I was just going to mention this, that what is emerging recently, there was vicious fights also about people that only believe in the vesicle transport, and then others that said, no, you have these very large proteins that don't fit in the vesicle, so you have to have external maturation. And there's a lot of papers of the two sides claiming that they are right, and what is turning out, like so many times in biology, is that probably both things are happening.
Yeah.
This is a very complicated structure, so you get some cisternal maturation, most probably, but there's no doubt that there is vesicle shuttling. So it's kind of pointless when you look from a distance. Why are people fighting so much? Because it's, what is important is that I think this is a very important concept, right? So that whatever is the mechanism, that these different compartments of the Golgi, they concentrate enzymes that do a specific modification, and then the protein moves to the next one. How does it move to the next one? The first is right. And this is the example, right? This means that this is possible to do biochemically. So this means if you have a sequence of glucosidases, and then mannosidases, and then N-acetylglucosamine transferases, and then you can start dissecting how many sugars have been added and when, because of these very useful enzymes. For example, EndoH is an enzyme that only works on proteins that have already acquired this complicated carbohydrate structure in this step 4 here. So if you just then remove one of these sugars with mannose, it starts being EndoH resistant. So that's why people use this as kind of a readout of the step of maturation if the protein is approaching. And then maybe one thing that is important to remember, what we call a complex carbohydrate. In many cases, these complex carbohydrates have at the end sialic acid. And the reason sialic acid is important is because it's very negatively charged. And we discussed this, right? This is why it's very hard for a glycoprotein to flip on the membrane, because many of them are finished up with sialic acid, and that is very hydrophilic. It reacts with water, and it doesn't react with hydrophobic lipid bilayers, so repels. And here we are. This means this is exactly what George was asking. The two models of Golgi maturation that still not completely settled, but now the textbooks, when you read in Albert's, they are saying that probably what's going on is a combination of the two models. So it was kind of a waste of energy to fight so viciously over which model is correct. There's another fight in the Golgi which is amusing, which is that what is very interesting about the Golgi is similar to the nuclear membrane, right? We looked at in mitosis, the nuclear membrane falls apart, and then it has to be reformed. So same thing with the Golgi. In mitosis, the Golgi disappears. So there's a big discussion if a little seed of Golgi remains, and from that little seed, the Golgi grows back, or if a completely new Golgi has to be produced out of the yard. There's evidence of both things, and I think again it's kind of emerging that it's both, that you can have a seed, but because if you eliminate the seed, you can make a new Golgi.
When you say seed,
Kind of a remnant, a very small, sometimes just one small cisterna, then regenerate the others. So this fight was between Grant Warren lab, Golgi lab for a long time, and Jennifer really watched shorts, which you're gonna watch one of her videos. So she was very much focused on imaging, and she kept saying that she didn't believe that she couldn't see anything remaining after the closes. She saw just a lot of flow coming out of the yard. So she said there's no residual Golgi. And Grant Warren, biochemically, he thinks that he sees evidence for residual Golgi. Again, I'm not sure how important it is, but this is very typical that scientists get very specialized, and then they really care a lot about the details. But the important thing is to look from a distance, even things that we don't understand, and try to define which are the things that we have to understand, which are incremental details, and which are fundamental questions that are going to make a difference.
you said the lady we were watching works at Golgi Lab.
If you watch what?
The lady that we're going to be watching works at Golgi Lab.
She does everything. Jennifer is a very interesting. She's here actually in the area. She's at the NIH. She just moved to Geneva Farms. So she has worked on practically every step of her main contribution is that she was one of the first ones to do live imaging with fluorescent markers. So she basically looks at cells in which she's expressing fluorescent proteins. She was one of the first ones to use GFP right after it came out. So you're going to be hearing from her when we talk about imaging techniques. Anybody else questions about this one? Because next Thursday, we're going to have again a little graded exercise that what I'm going to be looking for is if these concepts are clear. So what are the proteins that are important on this very important transition, right? So some people mentioned in the questions it wasn't clear what I was asking. And when I asked what type of mechanism is involved in the transition between one organelle and the other one. So what I meant was budding and fusion. And then we can break down the budding and fusion into budding and tethering and fusion. So you have to understand the sequence and which type of molecule does both. But don't say that snares are cold proteins that are budding. And this is very important because snares now, it really became universal, and there's no known fusion event that does not involve snares target. It's a little like DNA. Anything else? Okay, so what I would like to do, let's see how much time we have left. What I would like you to do with your groups, right? So most of you watched the Randy Shankman videos. So I would like each group to produce just one question that you would like to ask if you were watching him in person. So pretend that he's here, you listen to those two talks, and I just want you to work together. This is not graded, so don't worry about it. So just be creative, something that formulate a question. And it doesn't have to be one per person, one per group. Something you did not understand or something you think is interesting in the future. Anything like that.
I'm interested in the different diseases, cancer disease and craniofacial disorder.
Any questions? questions yet?
Just have to look up this video.
Oh, there was that part about the test used in the second video where he was explaining like the method or something. That part confused me. But
Yeah.
that's about it. That was better than the other video. Yeah.
But he doesn't go like in depth with it.
Because there are temperature resistant, proteins like but I do know that say that protein can
Still, it took like 1 hour.
Yeah.
It's much more
One of like circle text.
That was more.
why we do it. No, I'm just saying. Let's switch things out, you guys.
Tired. 40 minutes. Did you like practice?
Yeah.
How does new like genetic or cell technology play into
Which one? This one? Oh, no. Okay, so
Falls apart.
yeah, I didn't get that
When
part.
like there are multiple.
And then nutrients. And then introduce like the nutrients which was able to
Yeah, I got the process out of like
I mean, when it goes in
Any ideas?
Oh, you didn't ask about the vesicle budding assay? I just didn't get that one at all.
What were we gonna ask? Just like the mechanism?
Yeah, or like how it works, what it is.
No, I meant like, yeah, like the question, like we're.
Let me see if I understand. There are proteins and molecules in the vesicles as well as Rab proteins are the total protein except they're not involved in direct fusion. Auxiliary portion, the helping portion. But it is, but it's actually the seg proteins like SAR1 that brings makes the fusion process. Right? Because I wrote here, the copy of SAR1.
we should do like he always wants to be that whole good thing or whatever, but like I just like
Yeah, I'm trying to get
I need to write it down.
These are pretty expensive. This is 50.
I mean, right now the only question we have is about the vesicle butting assay.
I only need one. So.
Yeah.
And I think we're just trying to raise more money for the competition than watch Jefferson.
Do you know who won it last time?
Who won it? I guess we're talking about collegiate national.
Maybe you could say, like, are there any new discoveries that would change your previous findings? A generic question.
Yeah, they didn't have more of a class.
You jumped too late.
Class, because only 2 people can contribute to each other class. There's like 4 guys ahead of me.
What Wi-Fi are you guys on?
Short,
Secure. Here?
I'm using my phone.
Wi-Fi's lately are really
Yeah, I'm always surprised. Seeing those like body types, right?
It'll like connect for a second for me and
I'm like,
then you can get my camera.
I don't want to Oh shit. Yeah,
Oh, you're trying to stay.
yeah, like.
Are you guys gonna be the first up there?
And your writing is like huge.
You need magnifying glass.
Well, okay. Oh my god.
Yeah. Oh yeah. That was pretty fun.
Oh my god, it's so crazy.
Yeah. I mean, I get to fit a lot of stuff.
Sarwan, you know where the part of
I have only like 1 hour. 2 to 3 to like search around like other data.
Oh, you're all booked up? Some professors are really nice and they'll like look at last one. That's what like a couple of honor seminars I had last year. It was Which one?
I think that's
You like hiking?
Somewhere huge, like outdoorsy somewhere.
Appalachian Trail? My friends did that.
Western Maryland,
Do you know they were just
such beautiful.
I have science.
My friends did the Appalachian Trail. Right?
Yeah, it's like 2,000
And you didn't do it all at once, so that was like the little caveat.
It's
It's
every
like,
trail club is trying to do the whole thing, but sectioning it out and so it's supposed to be
no,
That's like going quick.
yeah. Oh, I've done the whole thing. I mean, Appalachian Trail.
Yeah. I tried to get my company before to sponsor.
What are vesicle budding like? How do they work? Vesicle budding assays.
Was that in the video?
Yeah, he talked about it with the whole. There was that part with the test tubes and all that, and putting it down, but.
Oh, when you had this.
Yeah. Putting it down. That's not good.
PP or whatever, but it was, yeah. It was,
Yeah, he also talked about like,
it was
chemical,
LTC, there was like
using chemicals.
CEO.
Yeah. That part was for the.
I didn't know what that was.
That was like to get the temperature sensitive mutations, right?
Right, yeah. Trying to start
Yeah.
sure I got it.
Coming out
Oh,
the one, like, if you want, like, you could like work out something for like first or something.
It's like, well,
It's the sticker.
but you know, you shoot, right?
That was in the bio class
It's like,
for the union summit.
I was like,
Talking about cinematography, and he's like, I'm actually leaving my job as part of this editing team.
the transcript.
Showing some like a video.
So you can replace
He can I can make some voice for me, so now I'm just part of this team of like 12 people, and we're just getting signed up for like interact with other companies that want to do stuff. Some people do really boring stuff and want to like look commercials and stuff like that.
I was
But our
wondering,
team,
is it more like action stuff that you guys do? Or is it like,
we're working off with Red Bull.
yeah.
Yeah, they do really crazy stuff.
Yeah, so that's all you guys.
Yeah, like our first, I started working in the summer. There's 2 videos over the summer. One on paragliding in the desert.
What?
And the second one
You skip all the
extreme
we know that.
Unfortunately, I don't get to go there.
Okay. Still, look at all that footage, right?
People know that.
Red Bull.
Sec protein.
Yeah.
So it's not mutated, it's just that it's higher.
Yeah, so yeah, that was the one they introduced the mutation in, and when they raised the temperature, it stopped functioning, and so it was like budding. Pardon?
The protein.
Yeah, it's called at higher temperature.
But it still has the mutation.
Yeah, it was mutated even at like room temperature. It was like mutated all along. It's just when you put a lot of heat, it unfolds. It just makes it more unfolding. To unfold and gain heat. So when they raised the temperature, it denatured and it stopped functioning. So that's why they were able to see like while it was in the middle of the process of budding.
Yeah, so like from there I was thinking, did it have to be mutated for it to hold out?
Yeah, it did. Yeah.
Okay. Probably like some bond that was.
Yeah, I don't know how they did it exactly. It's pretty specific. Yeah.
Trampoline club. Actually do that.
They actually gave us something kind
I don't think we need like half an hour.
Yeah.
No. Our leash? Yeah. Sorry. We got a bunch of
Really specific and really
But it's weird because we have a bunch of like.
Okay, but if I can just write it down and leave it with us.
Oh, okay.
I need a
It's not rated.
Right on.
I like how you can see what the grade assignments are. It's a 5 at the end.
Really? Oh, that's
So I
funny.
knew we were going to have an assignment during English class.
Oh, okay. We'll memorize much next time.
Yeah, don't we have a quiz on the
I hear
card?
that. Yeah.
Yeah, she did.
Yeah, I'm just gonna download it the day before and have lunch.
What further left to go?
Rip.
Well, I wanted to ask about further treatment of CLS. It's very specific, specific disease, craniofacial
It was recommended.
recommended. There's no such thing about that. I would say. So for this disease, treatment for it is like it's focused around treating the symptoms instead of taking it or it's a genetic situation, but it's only one chromosome. So
Sorry, shitty question. What is the function of the vesicle building reaction and how does it work? And how does it work?
Very good.
Andrew, are you going to post this online?
Yes. Yes. I post all of them mostly same week, most of the time.
How do you, do you like send a link? Or is it on YouTube?
Yeah, I always do a BSCI 420 and then...
Okay.
It's on YouTube.
Yeah. Yeah. I have all 3 other lectures already on there. Okay. So.